Tillage, in agriculture, the preparation of soil for planting and the cultivation of soil after planting. Tillage is the manipulation of the soil into a desired condition by mechanical means. Tools are employed to achieve some desired effect, such as pulverization, cutting, or movement. Soil is tilled to change its structure, to kill weeds, and to manage crop residues. Soil structure modification is often necessary to facilitate its intake, storage, and transmission of water and to provide a good environment for seeds and roots. Elimination of weeds is important, because they compete for water, nutrients, and light. Crop residues on the surface must be managed in order to provide conditions suitable for seeding and cultivating a crop. If the size of the soil aggregates or particles is satisfactory, preparation of the seedbed will consist only of removing weeds and the management of residues. Unfortunately, the practices associated with planting, cultivating, and harvesting usually cause destruction of soil structure. This leaves preparation of the seedbed as the best opportunity to create desirable structure, in which large and stable pores extend from the soil surface to the water table or drains, ensuring rapid infiltration and drainage of excess or free water and promoting aeration of the subsoil. When these large pores are interspersed with small ones, the soil will retain and store moisture also. Seedbed preparation procedures depend on soil texture and the desired change in size of aggregates. In soils of coarse texture, tillage will increase aggregate size, provided it is done when only the small pores are just filled with water. Tillage at other than this ideal moisture will make for smaller aggregates. By contrast, fine-textured soils form clods. These require breakage into smaller units by weathering or by machines. If too wet or too dry, the power requirements for shattering dry clods or cutting wet ones are prohibitive when using tillage alone. Thus, the farmer usually attempts tillage of such soils only after a slow rain has moistened the clods and made them friable. Some soils require deepening of the root zone to permit increased rate of water intake and improved storage. Unfavorable aeration in zones of poor drainage also limits root development and inhibits use of water in the subsoil. Tillage, particularly conventional plowing, may create a hardpan, or plow sole, that is, a compacted layer just below the zone disturbed by tillage. Such layers are more prevalent with increasing levels of mechanization. They reduce crop yields and must be shattered, allowing water to be stored in and below the shattered zone for later crops. Tillage is also associated with the loss of fertile topsoil through erosion, a serious threat to the longevity of arable land. 271. Primary tillage equipment. Equipment used to break and loosen soil for a depth of 15 to 90 centimeters, 6 to 36 inches may be called primary tillage equipment. It includes moldboard, disc, and rotary, chisel, and subsoil plows. The moldboard plow is adapted to the breaking of many soil types. It is well suited for turning under and covering crop residues. There are hundreds of different designs, each intended to function best in performing certain tasks in specified soils. The part that breaks the soil is called the bottom or base. It is composed of the share, the landside, and the moldboard. When a bottom turns the soil, it cuts a trench, or furrow, throwing to one side a ribbon of soil that is called the furrow slice. When plowing is started in the middle of a strip of land, a furrow is plowed across the field, and on the return trip a furrow slice is lapped over the first slice. This leaves a slightly higher ridge than the second, third, and other slices. The ridge is called a back furrow. When two strips of land are finished, the last furrows cut leave a trench about twice the width of one bottom, called a dead furrow. When land is broken by continuous lapping of furrows, it is called flat broken. If land is broken in alternate back furrows and dead furrows, it is said to be bedded or listed. Different soils require different shaped moldboards in order to give the same degree of pulverization of the soil. Thus, moldboards are divided into several different classes, including stubble, general purpose for clay and stiff sod soil, slit, black land, and chilled general purpose. The black land bottom is used, for example, in areas in which the soil does not scour easily, that is, where the soil does not leave the surface of the emerging plow clean and polished. The share is the cutting edge of the moldboard plow. Its configuration is related to soil type, particularly in the down suction, or concavity, of its lower surface. Generally, 3 degrees of down suction are recognized: regular for light soil, deep for ordinary dry soil, and double deep for clay and gravelly soils. In addition, the share has horizontal suction, which is the amount its point is bent out of line with the landside. Down suction causes the plow to penetrate to proper depth when pulled forward, while horizontal suction causes the plow to create the desired width of furrow. 272. Moldboard plow bottom sizes refer to width between the share wing and the landside. Tractor plow sizes generally range from 10 to 18 inches, 25 to 45 centimeters, although larger, special purpose types exist. Secondary tillage. Secondary tillage, to improve the seedbed by increased soil pulverization, to conserve moisture through destruction of weeds, and to cut up crop residues, is accomplished by use of various types of cultivators, harrows, rollers, or pulverizers, and tools for mulching and fallowing. Used for stirring the soil at comparatively shallow depths, secondary tillage equipment is generally employed after the deeper primary tillage operations. Some primary tillage tools, however, are usable for secondary tillage. There are 5 principal types of harrows: the disc, the spike tooth, the spring tooth, the rotary cross harrow, and the soil surgeon. Rollers, or pulverizers, with V-shaped wheels make a firm and continuous seedbed while crushing clods. These tools often are combined with each other. When moisture is scarce and controls of wind and water erosion necessary, tillage is sometimes carried out in such a way that crop residues are left on the surface. This system is called trash farming, stubble mulch, or subsurface tillage. Principal equipment for subsurface tillage consists of sweeps and rod weeders. Sweeps are V-shaped knives drawn below the surface with cutting planes horizontal. A mounted set of sweeps provided with power lift and depth regulation is often called a field cultivator. The typical rod weeder consists of a frame with several plow-like beams, each having a bearing at its point. Rods are extended through the bearings, which revolve slowly under power from a drive wheel. The revolving rod runs a few inches below the surface and pulls up vegetative growth. Clearance of the growth from the rod is assisted by its rotation. Rod weeders are sometimes attached to chisel plows. Some control of weeds is obtained by tillage that leaves the middles between crop rows loose and cloddy. On mechanized farms, this is often accomplished with a cultivator pulled by a tractor. When a good seedbed is prepared only in the row, the seeded crop can become established ahead of the weeds. Plowing with the moldboard plow buries the weed seeds, retards their sprouting, and tends to reduce the operations needed to control them. If weed infestations become bad, they can be reduced somewhat by undercutting. 273. Minimum tillage. The use of cropping systems with minimal tillage is usually desirable, because intensive tillage tends to break down soil structure. Techniques such as mulching also help prevent raindrops from injuring the surface structure. Excessive tillage leaves the soil susceptible to crusting, impedes water intake, increases runoff, and thus reduces water storage for crop use. Intensive vegetable production in warm climates where 3 crops per year may be grown on the same land may reduce the soil to a single grain structure that face cementation and poor aeration. The loosening and granulating actions of plowing may improve soil structure if the plowing is done when the moisture content is optimal. If not so timed, however, plowing can create unfavorable structure. The lifting and inversion of the furrow slice likewise may not always be desirable, because in many cases it is better to leave a trashy surface. The concept of minimum tillage and no-till agriculture has received much attention. One type of minimum tillage consists in seeding small grain in sod that has been relatively undisturbed. Narrow slits are cut in the sod, and seed and fertilizer are placed in the breaks thus formed. Soil normally subject to erosion can be planted to grain this way while still retaining the erosion resistance of the sod. The technique has been successful in preparing winter grazing in southeastern portions of the United States. In another type of minimum tillage, the land is broken and planted without further tillage in seedbed preparation. One approach involves breaking the land and planting seeds in the tractor tracks, wheel track planting. The tractor weight crushes clods and leaves the seed surrounded by firm soil. Another method consists of mounting a planter behind the plow, thus planting without further traffic and leaving a loose seedbed that is satisfactory in areas where post-planting rains may be heavy. In some areas, where winter rain often comes after wheat is drilled, a rotation of wheat following peas has been successful. After the peas have been harvested, the field is rough plowed, and fall wheat is then drilled indirectly. All these methods minimize expense and land preparation, tending to leave the soil rough, which reduces erosion and increases water intake. Somewhat similar systems are employed with row crops, where chemical weed control assists in reducing need for cultivation. 274. Mulch tillage. Mulch tillage is a system in which crop residues are left on the surface, and subsurface tillage leaves them relatively undisturbed. In dryland areas, a maximum amount of mulch is left on the surface. In more humid regions, however, some of the mulch is buried. Planting is accomplished with disc openers that go through several inches of mulch. Since mulch decomposition may deprive the crop of nitrogen, extra fertilizer is often placed below the mulch in humid areas. In rainy sections, intercropping extends the protection against erosion provided by mulches. Intercrops are typically small grains or sod crops such as alfalfa or clover grown between the rows of a field crop that reach maturity shortly after the field crop has been established and furnish mulch cover for a long time. If growth of the intercrop competes with the main crop for moisture and nutrients, that growth may be killed at seeding time or soon thereafter by undercutting with sweeps. Tillage in dry areas must make maximum use of scanty rainfall. The Lister double moldboard plow, or middle breaker, is here used to make water impounding ridges that promote infiltration. 12.4.3 Planting, harvesting date manipulation. The availability of crop varieties with a range of maturity periods allows considerable flexibility in planting dates. The most common manipulation of planting dates is the utilization of safe dates, which are a range of dates where a given crop can be planted such that it is in a relatively invulnerable stage when the damaging stage of the pest is present. For example, delaying the planting of winter wheat till fly-free dates in the fall reduces damage of Hessian fly, Myetiola destructor. Alternatively, planting can be timed so that the vulnerable stage of the plant coincides with periods when alternate hosts are abundant or weather conditions are likely to be unsuitable for pests to exploit the crop. The use of safe planting dates was an important form of pest control before the advent of chemical insecticides. In light of the technological advances that allow more accurate predictions of pest and crop phenology, the use of this safe and inexpensive alternative deserves reconsideration. 275. Limiting the length of time that a crop is exposed to insects can provide a useful alternative insect management strategy. Short-season cotton varieties have been adopted widely across the U.S. with the aim of avoiding insect problems by producing an early maturing crop. 12.4.4 Sowing plant density. Sowing density determines the amount of light available for plants, affects the efficiency of photosynthesis, and has an impact on the plant nutrition area. Plant spacing and shading can be an important influence on the population growth of some pests. Corn earworms prefer open plant canopies. They therefore most readily infest late planted and wide row soybeans. Soybean canopies will close sooner if planted in narrow rows and can help reduce an infestation. Similarly, planting young spruce trees as close together as possible can achieve early closing of the canopy and can suppress infestations of a spruce weevil, the Lacetus canis, which seem to require open sunlit areas for survival. Thinner and less healthy stands of grain fields tend to be attacked by the chinch bug. Because canopy shading is apparently not preferred by the pest, chinch bug management recommendations often include practices that encourage thick, vigorous stands. 12.4.5 Intercropping. Intercropping is a method of increasing agricultural productivity by planting 2 or more crops simultaneously on a specific plot of land in a specific row pattern. The primary goal of this type of cropping is to make use of the space between 2 rows of main crop and produce more grain per unit area. Small farmers who depend on rain for higher production are more likely to use it. This procedure has a specific row pattern, such as 1, 1 or 1, 2, which means that the primary crop is in the first row and the other crops are in the second or third row. These crops are blended in this technique despite having different nutrient needs. It guarantees the best possible use of the nutrients given. 276. Additionally, it stops pests and diseases from spreading to every plant involved in a particular crop. Principles of intercropping. Crops grown in tandem should have complementary rather than competitive effects. The subsidiary crop should be of shorter duration and faster growing habits to take advantage of the main crop's early slow growing period, and it should be harvested when the main crop begins to grow. Agronomic practices for the component crops should be similar. Erect growing crops should be intercropped with cover crops such as pulses to reduce or control soil erosion and weed population. This also aids in reducing water evaporation from the soil's surface. The root depths of the component crops should be different so that they do not compete for nutrients, water, and root respiration. A standard plant population of the main crop should be maintained, whereas the plant population of subsidiary crops can be increased or decreased depending on the situation. Component crops infested with similar pest and disease pathogens and parasites should not be chosen. The planting method and management should be simple, less time-consuming, less cumbersome, economical, and profitable in order to be widely adopted. Types of intercropping. Strip intercropping. Term, strip cropping, refers to the practice of cropping different cultures in strips. To prevent soil erosion, it is a common practice on sloped terrain. Strip cropping in agriculture also applies to even terrains. The strips are sufficiently narrow to generate agronomic interactions between neighboring crops. Strip width is therefore essential to the design and operation of this cropping pattern. The strips are frequently changed from year to year. Basically, strip cropping agriculture is utilized to either enhance the growth of primary species or improve soil health. Parallel cropping. Parallel cropping is the practice of growing crops that have different natural behaviors but no competition. An example of parallel cropping is wheat and mustard. After planting wheat, farmers typically broadcast mustard in the same field. It makes greater use of resources like light, nutrients, and moisture. The productivity of the 277 system is hampered by inefficient moisture usage and competition between the various root systems of the crops cultivated together. Synergistic cropping. Synergistic cropping is when two crops are grown on a unit area at the same time and produce more as a whole than they would if they were grown separately on a unit area basis. The concept of synergistic cropping first emerged in the 1980s. This technique is based on agronomic approaches primarily targeted at improving soil fertility. As a result, the overall health of the soil microorganism plant system is focused, rather than on production alone. Multi-story cropping. Multi-story cropping is the process of cultivation of more than two crops at once on the same plot of land with varying heights. Examples: coconut, pepper, cocoa, and pineapple. The crops are cultivated together on the land, utilizing land, water, and space in the most effective and inexpensive ways possible. Plantings having an overstory of trees or shrubs and an understory of specialized or agronomic crops or pastures are referred to as multi-story crops. There is enough space between trees to allow forage or crops in the understory to receive enough sunlight. Multi-story cropping is sometimes known as forest farming. Native forest tree canopies would be controlled to permit the cultivation of such crops or forage. Relay intercropping. Relay cropping is a type of cropping arrangement in which one crop is seeded into a standing second crop well before the second crop is harvested. Growing rice, cauliflower, onion, summer gourds is an example for relay intercropping. Relay cropping has the potential to resolve a number of conflicts, including inefficient use of available resources, disagreements over sowing time, fertilizer application, and soil degradation. Relay cropping is a complex set of resource-efficient technologies capable of improving soil quality, increasing net return, increasing land equivalent ratio, and controlling weed and pest infestation. Relay planting has less risk because it does not require relying just on one crop. Relay cropping is also known as overlapping cropping. Alley cropping. Alley cropping is one of several practices that deal with trees and crops growing on the same plot of land. Alley cropping is a method of growing trees or shrubs and agricultural crops in alternate rows. Trees are frequently pruned to reduce the shading of agricultural crops. Alley cropping can help with nutrient cycling and erosion control as well. Alley cropping can thus be regarded as recreating a savanna structure in an Agro 278 forestry system with many canopy layers, including an overstory of nut trees, a mid-layer of fruiting small trees and shrubs, and a ground cover of annual crops or perennial grass ground cover. Row intercropping. Plants are arranged in rows in this case, as the name suggests. Cereals and legumes such as corn and beans are a common and beneficial combination. Row ratios can range from single to multiple rows. Row cropping provides additional nitrogen fixation by legumes in symbiosis with bacteria of the Rhizobium genus. Temporal intercropping. Combined plants in this intercropping method require different maturing times. When the fast-growing plant is harvested, the slow-growing plant is given more room to grow. Mixed intercropping. Practice of intercropping entails sowing different species, two or more, in the same terrain with no distinct arrangement in rows or in the same rows. In this case, the sowing and harvesting seasons coincide. Mixed cropping protects the primary culture from winds, frosts, droughts, and other extreme weather conditions. 12.4.6 Trap cropping and irrigation. The intercropping technique, as the name implies, aids in pest trapping to protect the main culture. Among the most common trapping plants are mustard and marigold. The basic concept is to attract insects or fungi to the sacrificial secondary crops, protecting the cash crop. Blue Hubbard squash is said to be effective against squash bugs, squash vine borers, and spotted and striped cucumber beetles. Trap intercropping allows for pesticide savings by requiring no or only partial chemical application to trapping areas. Guard cropping. Guard crops are thorny or tough plants that grow around cash crops or along field edges. Crop guards are used as barriers to keep the main species safe from winds and invasions. As a result, sorghum is grown next to cotton and safflower next to chickpea. 279. Repellent intercropping. Farmers use pest repellent plants as a sustainable pest management technique when using this intercropping method. It is based on the repellent effect of specific species, which protects the cash crop. The repellent keeps insects away from their host plant, as in the case of planting leeks to keep bean flies away from beans. Push-pull cropping. For the sake of the cash crop, the intercropping practice combines both trap and repellent plants. While trap species attract or pull pests, repellent species repel them. Growing Napier grass to pull and Desmodium legume to push to protect corn from stem-boring corn larvae is an example of this technique. 12.5 Summary. Pest control, a process that maintains nuisance organisms below economic thresholds. Agricultural infestation results in widespread deterioration of biodiversity, with important implications for pest control. Pest control is a program to eliminate pests from fields to practice good hygienic practices. It could include many of the measures to prevent entry, harborage and infestation of pests. Moreover, it should be effective enough to provide a means to monitor, detect, and eradicate pests. Different techniques work for different pests. Most of the pests can be controlled by intercropping the fields without using any chemicals, like insecticides, pesticides, etc. Many genetically modified organisms, some agricultural methods and organic oils can prove really beneficial in biological or natural pest control. Natural. Botanical versus synthetic pesticides. Natural compounds called botanical pesticides are taken from plants with known insecticidal characteristics. Botanical pesticides are being used as vital alternatives to synthetic pesticide chemicals because the bulk of them pose environmental risks. Botanical insecticides were regarded as vital crop protection techniques before the development of the synthetic pesticide DDT. Initially, the usage of botanical insecticides was obsolete due to the emergence of low-cost, extremely efficient synthetic insecticides. However, a number of issues with synthetic pesticides, including environmental contamination, residues in food and feed, and insect resistance, have brought them to the forefront once more. Several commercial solutions manufactured from plants like neem, Azadirachta indica, which has demonstrated good effectiveness as a botanical pesticide, are already available on the market. As insects and other organisms developed resistance to synthetic pesticides throughout time, nickel pesticides have grown in popularity relative to chemical pesticides. Chemicals like nicotine and oils like citrus oils are among the insecticides found in plants. These natural pesticides are said to create insecticides that are quick to kill insects and have little to no negative impact on the environment. Why should biopesticides be used instead of synthetic pesticides? Biodegradable. Less toxic to humans. 283. Act quickly. Non-pollutant. Easily available in environment. Advantages of botanical pesticides over synthetic pesticides are listed below. Table 1. Comparison of botanical versus synthetic pesticides, Lingai et al. 2020. Due to their numerous benefits and drawbacks, botanical pesticides should only be used as alternatives in IPM programs and in conjunction with other available control techniques. 13.3 Formulations and insecticide toxicity. Toxicity is ability to pesticide to cause illness in pests. For testing toxicity, animal is exposed to different degrees of pesticides formulated product. The types of toxicity include acute. In this exposure is for shorter duration. It is chemical ability to cause illness to a person on single exposure. Main route of inhalation includes dermal, skin, inhalation, lungs, oral, mouth, and eyes. 284. Chronic. It occurs after long-term exposure to harmful pesticide. In this small-term exposure is repeated several times. Effective chronic exposure includes birth defects, nerve disorder, tumor production, etc. Pesticide toxicity is either measured as LD50, lethal dose, or LC50, lethal concentration. LD50 is amount of chemical that causes death in 50% animals on exposure to oral or any other exposure. LC50 is the concentration of a chemical in inhalable form that is lethal to 50% of test organisms. Its units are milligrams of substance per kilogram of test animal body weight, milligram per kilogram. Effective LD50 and LC50 includes. Even a low value of toxicant can cause high toxicity. They are based on single dose or short-term acute exposure. LD50 does not include combine effect leading to carcinogenic effects. Ref. Understanding pesticide risks, toxicity and formulation, 2016. Formulation of insecticides. Formulation makes the toxicant easier to use, and more effective through the addition of emulsifiers, wetting agents, stickers, spreaders, etc. Importance of pesticide formulation. Pesticide formulation lowers the risk of people and other non-target organisms. Pesticide application should be specific as given on pesticide labels. 285. Applying pesticides in greater amount is illegal and not cost-effective. It may lead to risk on non-target species. Methods of pesticide formulation includes baits. They are target-specific and useful for social insects like ants and termites. They contain food products to attract a specific group of pests. Dusts. Dust is dry and fine powders. Applied in case sprays are harmful for plants in indoor space. Pests get harmed either through absorption or ingestion during grooming. Protective masses should be wearied against them. Granules. These particles are larger in size than dusts. They are applied to soil to control weeds, nematodes, and soil-dwelling insects. In this active ingredient is released slowly and may require moisture or soil incorporation. Sprays. They come as wettable powder, flowable, emulsifiable concentrate, sprayable oils, and oil-based sprays. They are easily absorbed through skin, flammable and corrosive. Aerosols. They are repurchased in pressurized containers in a ready-to-use form. 13.4. Botanical pesticides. Botanical insecticides are substances that contain dried, pulverized plant material, unprocessed plant extracts, or compounds that have been extracted from plants and are used to kill or repel insects. They are made of organic and natural components and are natural pesticides. These work well against various organisms such as viruses, nematodes, bacteria, fungus and insect pests. Classification of pesticides. Based upon target pests, the mode of action, and the chemical makeup of pesticides can be categorized as A. Synthetic pesticides. There are 4 subgroups of synthetic pesticides. B. Biopesticides. These are broken down into 4 subgroups: pheromones, plant-incorporated protectants, microbial biopesticides, and botanical pesticides. The botanical pesticides are the main topic of this chapter. 286. Table 1. Different types of pesticides. Pesticides. Synthetic pesticides. B. Biopesticides. Organochlorines, microbial biopesticides, organophosphates, plant-incorporated protestants, carbamates, botanical pesticides for thrins, pheromones. Classification of botanical pesticides. The biopesticides are divided into different categories based on kind, active ingredient, crop type, and area. It is separated into insecticides, fungicides, and herbicides based on kind. Nicotine, pyrethrin, rotenone, matrine, and azadirachtin are some of them. Table 2. Botanical pesticides on the basis type, active substance and crop type can be divided as type by active substance, crop type. Insecticides: nicotine, oilseeds and pulses; herbicides: pyrethrin, vegetables and fruits; fungicides: rotenone, turfs and ornamentals; matrine. Azadirachtin. Advantages of botanical pesticides. The active ingredients' quick decomposition is advantageous. Products can be utilized right before harvesting. A lot of these products work fairly rapidly to stop insects from feeding, even though they don't kill insects over time. Since most of these products have a stomach action and are quick decomposition, the majority of these compounds may be more selective to insect pests and less aggressive with natural enemies. 287. Not phytotoxic. Resistance to these substances does not spread as quickly. Less damaging to the environment and less environment load, eco-friendly. Affect only one pest, target specific. Biopesticides work against pests in little dosages. Biodegradable and non-persistent. Disadvantages of botanical pesticides. They are not truly insecticides since many are merely insect deterrents. Slow effect. Not all natural insecticides are safer for other animals to consume. They might not be accessible all year round. Lack specified tolerances for residues. There are no legal registrations establishing their use. Not every advice given to growers has been backed up by research. It causes neurological, psychological, behavioral dysfunction and diseases including cancer, blood disorder in humans. They harm the environment by destroying marine life, reducing biodiversity, and other things. Insects may quickly adapt a resistance to them. Repression of predators and parasitoids. Negative impacts on organisms that are not the target and loss of soil fertility. Mode of action. 288. 289. The exact biochemical interaction that a pesticide uses to achieve its impact is referred to as its mode of action. The mode of action typically includes the particular enzyme, protein, or biological process impacted. Mode of action particularly relates to which biological process the pesticide inhibits, unlike the majority of other classifications, which focus on the pest's controlled, physical qualities, or chemical makeup. 13.4.1 Pyrethrins. Structure. Cenerin IC. Source. It is one of the most significant botanical pesticides used in India. It is made from flower of Chrysanthemum cinerariifolium. Pyrethrum is concentrated heavily in a flower's blooms. 6 esters are produced when the acids chrysanthemic and pyrethric and the alcohols pyrethrilone. Biopesticides. Rotenone. Sabadilla. Pyrethrins neem. Nicotine. Cinerolone and jasmolone are combined. These chemicals are derived from this plant and have been shown to have known insecticidal activity. 6 active ingredients: pyrethrin I, pyrethrin II, cenerin I, cenerin II, jasmolin I, and jasmolin II, combine to form pyrethrum. The esters of chrysanthemic acid are pyrethrin I, cenerin I, and jasmolin I, while the esters of pyrethric acid are pyrethrin II, cenerin II, and jasmolin II. The ratio of pyrethrins, cenerins, and jasmolins in a typical pyrethrum extract is 10:3:1. In terms of concentrations, pyrethrins are the most prevalent form of active components when compared to cenerins and jasmolins. Pyrethrin structure. Major target pests of pyrethrins are mosquitoes, sawfly larvae, caterpillars, leafhoppers, aphids, and beetles, Chilicoides, Variopenis, coquillette, houseflies, and flower beetle. Mode of action. It affects both central nervous system and peripheral nervous of insects. It prevents the process of sodium-potassium ion exchange in the nervous system of insects. It prevents the process of sodium-potassium ion exchange in the nervous system of insects. The insect get paralyzed as a result. Pyrethrum pesticides target the insect's nerve cells, causing paralysis and ultimately death. Mechanism of action. Impact CNS. Prolonged deactivation of sodium channels that are voltage gated. Prolonged fibers excitation. Greater selectivity with additional neurotoxins. 290. Source: Louise N. Ethier, 2016. Pyrethroids, mode of action on neurons. Disadvantages: many insects can metabolize pyrethrins, thus after a brief time of paralysis, they recover instead of dying. Therefore, piperenal butoxide (PBO) is combined with pyrethrins to impede metabolization and recovery from poisoning induced by pyrethrins alone, which results in insect death. Pyrethrins are less effective without PBO. Toxic effects on mammals: although pyrethrins are not poisonous to humans, they do have an impact on cats. When consumed, they are not easily absorbed by the digestive system and are instead quickly hydrolyzed in the liver's alkaline environment and the gut's acidic environment. Pyrethrins become harmful when inhaled rather than when they were consumed directly. High-dose inhalation may result in headaches, nausea, vomiting, diarrhea, and other nervous system abnormalities. Pyrethrum dust exposure can induce allergic responses or skin rashes. Ongenetic changes or birth problems are not brought on by prolonged exposure to pyrethrins. There are no current treatments for its toxicity. 13.4.2 Rotenone. Structure: C23H28O6. It is isoflavonid chemically. 291. Rotenone molecular structure. It is made from the tropical plants Tephrosia virginiana, Lonchocarpus utilis, Lonchocarpus urucu, and the bean Daris elliptica, Daris involuta. Both Daris and Lonchocarpus provide 13% of the rotenone, while Lonchocarpus spp. is indigenous to the Western Hemisphere, namely South America. Daris spp. is indigenous to the Eastern tropics, such as Asia. Roots and stems of plants are used to make rotenone extract. It is a plant insecticide with a broad spectrum. It can be sprayed on plants or used as root powder. It is derived from cube root in acetone or ether. Following extraction, 2 to 4% of rotenone resins which contain a number of similar but less insecticidal chemicals known as rotenoids are formed. Cube root can also be dried, ground into a powder, and combined with an inert binder to create an insecticidal dust. Mode of action: the active ingredient is rotenone, which serve as a contact in food poison, cellular respiratory enzyme inhibitor, stomach poison. Rotenone, a potent inhibitor of cellular respiration, causes nutritional compounds to be converted into energy at the cellular level. Rotenone is a substance that can be ingested and used as a repellent. Inhibiting electron transport at the mitochondrial level blocks the phosphorylation of ADP to ATP, which in turn prevents the metabolism of insects. Rotenone poisoning in insects causes symptoms such as a decrease in oxygen consumption, respiratory depression, ataxia, convulsions, and ultimately paralysis and respiratory arrest. Rotenone poisons the nerve and muscle cells in insects, forcing a swift end to feeding. Insect death happens several hours after chronic exposure. 292. It is extremely poisonous to fish and is employed in water management programs as fish poison pesticide. It is created by PBO, piperenal butoxide. Other effects of rotenone include elevated level of nitric oxide and malondialdehyde, global cellular impairment, accumulation of alpha-synuclein and polybiketine, activation of astrocytes and microglial cells, neuroinflammatory responses, glutamate excitotoxicity, oxidative stress, abnormalities in microtubule assembly and spindle formation during mitosis. 293. Toxic effects on mammals. Its direct contact leads to skin irritation and irritation of mucosa membrane. Its chronic exposure leads to liver and kidney damage. It may lead to tumor formation, but until now it has been considered as non-carcinogenic. Commercial rotenone has not shown hazards to human beings. It is a strong cell poison that mammals detoxify with the help of their liver enzymes. Rotenone is also more poisonous when inhaled than pyrethrins are. Rapid breathing, muscle tremor, nausea, and vomiting are symptoms of excessive rotenone exposure. The strong dose results in convulsions followed by death from circulatory collapse and respiratory paralysis. 13.4.3 Sabadilla. Structure: Sabadine C32H49O9. Veratridine C36H51O11. Source: it is made from ripe Shanocolin officinale seeds, Liliaceae. Central and South America are the two continents where this tropical lily plant is found. This plant's seeds have been found to contain significant amounts of the alkaloids that give it its toxic effects. It comes from Venezuela and contains schistocyanidylene. Several alkaloids are produced when its seeds are roasted and aged. Alkaloids are substances that have physiological effects and are found in many plants. They belong to the heterogeneous class of cyclic compounds, and their rings contain nitrogen. Caffeine, nicotine, cocaine, quinine, and strychnine are some of its alkaloids. Veratrine alkaloids are the name given to its alkaloids. They make up 3 to 6% of ripe, old seeds. Mode of action: it damages the neuron cell membrane, which results in decreased nerve activity, paralysis, and eventual death. Toxic alkaloids in insects alter the function of the nerve cell membrane, leading to a loss of nerve function, paralysis, and insect death. Toxicity to mammals: contrary to the ground seeds, which are one of the plant insecticides with the lowest animal toxicity, the isolated alkaloids of these plants are among the most toxic and skin irritants. It lasts for a long period, has lingering effects in sunshine, and dissolves quickly. It 294. Also has a smooth toxic effect. By interfering with the functioning of the sensory, motor, and respiratory nerves, the insect is paralyzed and dies. Caterpillars, leafhoppers, thrips, stink bugs, and squash bugs are all susceptible. It appears like dust and is made from ground seeds. Pure alkaloids from it are exceedingly harmful. It has a strong sneezing-inducing action and is exceedingly irritating to the skin and mucous membrane when inhaled. It can affect circulation and cause headaches, nausea, vomiting, and diarrhea even at tiny doses. An excessive amount may cause convulsions, heart paralysis, and breathing failure. Their alkaloids can penetrate the mucosa membrane and be absorbed. 13.4.4 Nicotine. Structure: C10H16N2O4S. Source: it is a fundamental alkaloid generated from tobacco, Nicotiana tabacum, which is found in a few Solanaceae plants. It has 2 to 8% dried tobacco leaf content. It has 40% nicotine sulfate in it. Mode of action: in both mammals and insects, it is a rapidly acting nerve poison. It competes with the acetylcholine receptor by binding with it and causing controlled nerve firing. The postsynaptic membrane's principal neurotransmitter, the cholinergic receptor, interacts with acetylcholine and changes the membrane permeability. The physiological systems that depend on nerve input for optimal operation are quickly rendered incapable of operating as a result of this interruption of regular nerve impulse activity. New nerve impulses are produced as a result of nicotine action, and these impulses result in convulsions and death. Neonicotinoids, and important. 295. New class of insecticides, are synthetic analogs or derivatives of the nicotine structure. These include, among others, imidacloprid, thiamethoxam, nitempiram, and acetamiprid. Mammalian toxicity: it is a class 1, most lethal poison in its pure form and highly poisonous to mammals. It can be fatal if inhaled or come into contact with the skin, eyes, or mucous membrane. Due to the liver's efficient detoxification, eating is less dangerous. Symptoms of nicotine poisoning include nausea, vomiting, excess salt, trembling, and ultimately death from circulatory and respiratory collapse. Rapid poisoning is frequently lethal. The only necessary treatment for nicotine toxicity is symptomatic and includes extended artificial breathing. 13.4.5 NEEM. Structure: azotaractin. 296. This substance is a tetraterpenoid found in the Meliaceae family, namely in the neem tree, A indica, a native of India. Source: it comes from the Azadirachta indica neem tree. On various continents, this tree can be found in arid tropical and subtropical climates. Bitter azadirachtin, its main ingredient, is both a feeding inhibitor and a growth regulator. The substance is present in the tree's bark, leaves, and fruits, although the concentration is highest in the seeds. Although this molecule hasn't been artificially created in the lab yet, pure samples of it have shown better outcomes in tests than extracts have. Salinine, meliantrol, and azotaractin are the 3 most notable chemicals in the extract, with the latter having the highest concentration. There are 18 different components in the extract. Numerous additional elements of neem, including ismeliantriol and salinan, are also active in different ways. Numerous additional elements of neem, including ismeliantriol and salinan, are also active in different ways. Its products include teas, dusts, and extracts from whole fruits, seeds, and seed kernels manufactured from leaves and bark. Its seeds can also be used to extract oil. Its oil suffocates insects and is more akin to vegetable or horticulture oil. Commercial neem preparations are available under a variety of brand names, including Neem Gold, Neemazole, Eco Neem, Neemark, Neemcure, and Azadine. Mode of action: it is a complex mixture of substances with biological activity. Neem is primarily effective in controlling eating behavior in insects, while it can also be a repellent, growth regulator, oviposition suppressor, sterilant, or poison. It serves as an insect repellent and stops insects from starting to feed. Insects stop feeding while using this detergent as a feeding agent. Neem is a growth regulator that is suspected to interfere with chitin formation, disrupting normal development. 297. 13.5 Synthetic organic insecticides and their mode of action. The synthetic insecticides have become primary agents for insect control. In general, they penetrate insects readily and are poisonous to a variety of species. Synthetic pesticides have negative impact on environment, health of animals and humans. Farmers heavily rely on synthetic pesticides to control insects in their crops, and their use grew ubiquitous after World War II. DDT was first synthetic organic insecticide in 1874. Environmental contamination and resistance. The synthetic insecticides remain essential in modern agriculture despite their environmental drawbacks. By preventing crop losses, raising the quality of produce, and lowering the cost of farming, modern insecticides increased crop yields by as much as 50% in some regions of the world in the period 1945-65. They have also been important in improving the health of both humans and domestic animals. Malaria, yellow fever, and typhus, among other infectious diseases, have been greatly reduced in many areas of the world through their use. But the use of insecticides has also resulted in several serious problems. Environmental contamination. Development of resistance in pest species. After adversely non-target organism because of their poisonous properties. Some pesticides are persistent and when used in large quantities they permeate the environment, whereas others are short-lived or are digested by the animals they are consumed by. When an insecticide is used, much of it enters the soil, and runoff from treated areas or direct application can contaminate groundwater. Chlorinated hydrocarbons including DDT, aldrin, dieldrin, heptachlor, and BHC are the primary pollutants in soil. They become associated with food chains. Predator birds like eagles, hawks, and falcons are 298. 299. Typically the most seriously impacted, and DDT's impacts have been linked to significant population decreases in these species. Another issue with insecticides is that some target insect populations have a tendency to become resistant when susceptible members are eliminated and the resistant strains that remain proliferate, maybe eventually making up the majority of the population. Numerous synthetic organic insecticides no longer work on hundreds of kinds of dangerous insects. Because of the problems associated with the heavy use of some chemical insecticides, current insect control practice combines their use with biological methods in an approach called integrated control. In this approach, a minimal use of insecticide may be combined with the use of pest-resistant crop varieties, the use of crop-raising methods that inhibit pest proliferation, the release of organisms that are predators or parasites of the pest species, the disruption of the pest's reproduction by the release of sterilized pests. There are many classes of synthetic pesticides. The main groups of synthetic organic insecticides consist of class examples area of effect. Organochlorines OC, DDT, toxaphene, dieldrin, aldrin reproductive, nervous. Endocrine and immune system organophosphates OP, diazinon, glyphosate, malathion central nervous system carbamates C, carbofuran, aldicarb, carbaryl central nervous system. Pyrethroids, synthetic pyrethroids SP. Fenpropanthrin, deltamethrin, cypermethrin. Poorly understood. Others like neonicotinoids, insect growth regulators, IGRs, pheromones, etc. 13.5.10 ORGANOCHLORINES. Structure. Organochlorines are organic molecules that contain chlorine. They were the first to be discovered as organic insecticides. DDT and its related compounds, methoxychloroquine, kelthane, BHC, lindane, toxaphene, and cyclodine insecticides are included, e.g. aldrin, dieldrin, endrin, myrex, chlordane, heptachlor, endosulfan. Commonly used. Lindane, aldrin, dieldrin, chlordane, benzene hexachloride, endrin, heptachlor, endosulfan. Banned pesticides. DDT, dieldrin, chlordane, aldrin. Mode of action. OCs were useful in agriculture during the Green Revolution, and they were even effective against malaria. However, their toxicity has been confirmed in birds and fish. Because of bioaccumulation, their degradation products were discovered in many ecosystems. 300. They have also been found in underground waters. As a result, OCs can spread far beyond the areas where they can be used. They work on insects by opening sodium ion channels in their neurons or nerve cells, causing them to fire at the same time. The insect dies as a result of a spasm. Effect of organochlorines. They have the ability to easily bioaccumulate in biota. They increase their concentrations biomagnifications as they move up the food chain. Food contaminated with organochlorines enters the human body, and the effects of long-term exposure can be seen. Mutagenicity, endocrine disruption, carcinogenesis, and central nervous or peripheral disorder are the most serious health effects of OCs. Because these compounds are more soluble in fats, they accumulate in fatty tissues and concentrate in organisms at the top of the food chain in a community bioaccumulation. Lindane is still used to control human lice, as are endosulfan and kelthane. Harmful effects of OCs. They have long half-lives ranging from months to years, and in some cases decades. They are toxic to humans and other animals, as well as most aquatic life. Even at low concentrations, they can have serious short- and long-term consequences. Furthermore, non-301. Lethal effects of some of these pesticides, such as immune system and reproductive damage, may be significant. They accumulate in human, plant, and animal fatty tissues. The majorities of them are drawn to fatty tissues and organs and accumulate significantly in animals like fish. This means that higher up the food chain animals such as birds of prey and human, can accumulate higher levels of pesticides than lower-level animals. Organophosphates. Structure. Organophosphates are the most common and versatile class of insecticides. They have a shorter half-life than OC and do not accumulate in fatty tissues. They have wide range of mammalian toxicity, e.g., malathion, parathion, diazinon, chlorpyrifos, acephate fumigants, e.g., DDVP, and systemic insecticides are among them, dimethoate, disulfodin, Ronel. They are organic derivates of phosphorus and are nerve agents and are highly toxic. Two widely used compounds in this class are parathion, malathion. Other examples are diazinon, nalid, methylparathion, and dichlorvis. Mode of action. They are especially effective against sucking insects that feed on plant juices, such as aphids and mites. The chemicals are absorbed into the plant by spraying the leaves or applying solutions impregnated with the chemicals to the soil, allowing intake through the roots. Organophosphates kill insects by inhibiting the enzyme cholinesterase, which is required for nervous system function. These are phosphorus-containing insecticides derived from one of the phosphorus acids. Organophosphates are effective at controlling insect populations because they inhibit the nervous system's function. They have a low level of residual activity. 302. All these are nerve poisons. They block the site of an enzyme, acetylcholinesterase, ACT, that breaks down and removes acetylcholine from the nerve synapse. Normally, nerve cells in humans or insects' brains or muscles send tiny electrical pulses down tendrils to the cell's end, where the pulse must jump across a gap known as a synapse to other nerve cells. ACT moves from one cell to the next and binds with new cells, sending the electrical pulse down the new cell. These insecticides, as well as nerve gas agents, keep the ACT from escaping from the new cell, preventing it from receiving any more impulses. Commonly used organophosphates. Monocrotophos parathion. Malathion. Cholopyrifos phosmet. Diazinon. Methylparathion. 303. Phenotrophin. Banadorganophosphates. Chlorpyrifos, banned in most countries but still used in India. Diazinon, banned in agriculture but used in households. Phenotrophin, banned in agriculture except locust control. Parathion, banned for used in fruit and vegetables. Monocrotophos, banned on vegetables. Carbamates. Structure. The carbamates are a class of insecticides that includes carbamyl, methimyl, and carbofuran. Carbamates are insecticides made from carbamic acid. They are comparable to OPS in terms of mode of action, biodegradability, low solubility in fat, and mammalian toxicity. Carbaryl 7 is the most well-known carbamate. It is extremely toxic to humans. Carbofuran, propoxyr, methamyl, oxamyl, aldicarb, methamyl, bendiocarb, and formetanate are some other carbamates. Carbamates have an advantage over organophosphates in that their inhibitory reaction with acetylcholinesterase is reversible. Chronic carbonate exposure causes less illness than chronic organophosphate exposure. They are rapidly detoxified and eliminated from animal tissues. It is thought that their toxicity is caused by a mechanism similar to that of organophosphates. They repel insects well, but they can 304 also be rapidly detoxified from mammalian tissues, making them less toxic to both animals and humans. Mode of action. Carbamates have properties similar to organophosphates, but they last in the environment for a shorter period of time. They are believed to be less toxic. Carbamates inhibit AKE by carbamoylating the serine moiety at the active site rather than phosphorylating it. Because this is a reversible type of binding, their toxicity is milder and lasts less time. Carbamates have low CNS toxicity because they do not penetrate the CNS to a great extent. Because it is easily hydrolyzed, carbaryl is not considered a persistent compound. It is widely used in agriculture, including home gardens, where it is typically applied as a duster solution, such as aldicarb temic, aminocarb metasyl, aprocarb, bagan, carbaryl, 7, carbofuran, and carbofuran-based products Firaxton. Neurotoxic mode of action of carbamate insecticides. The carbamates, like the OP insecticides, inhibit acetylcholinesterase by carbamylation of the serine moiety at the active site rather than phosphorylation, with the important difference being that this is a reversible type of binding. Because the carbamylated enzyme reactivates quickly in the presence of water, the symptoms are less severe and last less time. Furthermore, because carbamates do not effectively penetrate the CNS, toxic CNS features are not prominent in the event of poisoning. 305. Commonly used carbamates. Carbaryl. Methiocarb, thiodecarb, carbofuran. Banned carbamates. Aldicarb. Pyrethroids. Pyrethroid insecticides are a chemical class of active ingredients found in wide range of modern insecticides. Pyrethroids or insecticides are used to kill mosquitoes and other insects. Pyrethroids have been demonstrated to be extremely safe for human health and the environment. It comes. 306. From the dried and crushed flower heads of two aster species, Chrysanthemum cinerarifolium and Chrysanthemum cochineum. Natural pyrethrum degrades rapidly in sunlight. Pyrethrins are widely used to control pests such as mosquitoes, fleas, flies, moths, ants, and other pests. Pyrethroids are present in many commercial insecticides, including household insecticides, pet sprays, and shampoos. They typically last for 1 or 2 days in the environment. Pyrethroids are difficult for plant roots to absorb because they bind to the soil. Pyrethroids rarely enter groundwater and do not contaminate drinking water supplies. Characteristics of pyrethroids. Low toxicity to mammals and birds. High toxicity for fish. Require very low doses to kill insects. Fast acting. Effective against chewing insects. Bind tightly to soil and organic matter, dissolve very poorly in water. Commonly used pyrethroid. Allothrin. Resmethrin. Permethrin. Cyfluthrin S-phenvalurate. Mode of action. Pyrethrin stimulates the nervous system of insects that touch or eat it. This quickly leads to paralysis and eventually their death. Pyrethrins are frequently mixed with other chemicals to increase their effectiveness. Pyrethrins are generally non-toxic to humans and other mammals. However, if it gets on your skin, it can be irritating. When inhaled, it has been reported to cause respiratory irritation, runny nose, coughing, difficulty breathing, vomiting, and diarrhea. People who have been exposed to pyrethrins have reported some of the same symptoms as those associated with asthma. 307. Pyrethroids disrupt the voltage-gated sodium channels that keep the nerve cell membrane electrically charged in mammals. Pyrethroids bind to the sodium channel subunit and slow the rate of activation opening, as well as the rate of inactivation closing, resulting in a stable hyperexcitable state. Sodium channels then open at more hyperpolarized potentials and stay open longer, allowing more sodium ions to cross the neuronal membrane and depolarize it. Based on its sign and mechanism of action, pyrethroid has been divided into 2 types. Type 1T and Type 2 CS syndrome. Type 1 compounds only keep the channel open long enough to cause action potentials to fire repeatedly. Type 2 compounds keep the channels open for so long that the membrane potential ultimately becomes depolarized to the point at which generation of action potential is not possible. GABA-gated chloride channels are also bound and inhibited by type 2 pyrethroids. These differences in the time of opening of sodium channels are believed to be at the basis of the differences observed between the T and CS syndromes. Neonicotinoids. 308. Figure 1. Neonicotinoids. The most important new class of synthetic insecticides is neonicotinoids. Imidacloprid, thiamethoxam, clothianidin, thiacloprid, dinotefuran, acetamiprid, nitinpyram, and sulfoxaflor are main neonicotinoids. They are an insecticide class that is chemically related to nicotine. The neonicotinoids, like nicotine, act on specific receptors in the nerve synapse. They are much more toxic to invertebrates, like insects, than they are to mammals, birds, and other higher organisms. Neonicotinoid insecticides are water-soluble. They can be applied to soil and absorbed by plants. Neonicotinoids are a type of insecticide that works by affecting the central nervous system of insects, causing paralysis and death. According to research, neonicotinoid residues accumulate in the pollen and nectar of treated plants, posing a risk to pollinators. As a result of this, and their widespread use, there is major concern that neonics play a significant role in pollinator declines. Neonicotinoids are also persistent in the environment, and when used as seed treatments, they translocate to residues in treated plants' pollen and nectar. A songbird can be killed by as few as one seed. These pesticides pollute waterways and are extremely toxic to aquatic organisms. Imidacloprid was the first neonicotinoid. It has systemic as well as contact activity against a wide range of sucking insects such as aphids, leafhoppers, and whiteflies. This product can be sprayed on plants, but it is usually more effective when applied to the soil. Dinotephorin is another highly water-soluble neonicotinoid that is particularly effective against sap-feeding insects. List of neonicotinoid. Acetamiprid. Clothianidin dinotephorin. 309. Imidacloprid. Nidinpyrum. Thiocloprid. Thiomethoxam mode of action. Neonicotinoids act as selective agonist at nicotinic acetylcholine receptors knockers. Neurotoxicity was described for both target and non-target species, insect pollinators. Sublethal concentrations of neonicotinoids alter social bee behavior, reduce colony survival, and impair the honeybee's olfactory memory and learning capacity as well as orientation and foraging activity. They bind to and interact with the insect nicotinic acetylcholine receptor site in insects. When neonicotinoids bind to an insect binding site, their electronegative tip, which contains a nitro or cyano group, interacts with a specific cationic subsite of the insect's receptor. Protonated nicotinoids requires a cationic interaction to bind to a mammalian receptor. These insecticides are selectively more toxic to insects than mammals. Neurotoxic mode of action of neonicotinoid insecticides. Neonicotinoids, like nicotine, bind to or fill the nicotinic acetylcholine receptors, preventing neural transmission. Nicotinic acetylcholine receptors are found in cells of both the central and peripheral nervous systems of mammals. Acetylcholine, a neurotransmitter, activates nicotinic acetylcholine receptors. While low to moderate activation of these receptors causes nervous stimulation, excessive activation causes paralysis and death by blocking the ACR receptors. Acetylcholinesterase degrades acetylcholine in order to stop signals from these receptors. However, neonicotinoids cannot be broken down by acetylcholinesterase, and their binding is irreversible. 310. 13.6 Insect growth regulator IGR. Insect growth regulators IGRs are compounds that inhibit insect growth, development and metamorphosis. They prevent insects from reproducing. They are used as insecticide to control the population of harmful pests like cockroaches and fleas. IGRs include synthetic analogs of insect hormones like ecdysoids, juvenoids, non-hormonal compounds such as precocines, anti-JH chitin synthesis inhibitors. Advantages. Effective in small quantities. Economical. Target-specific and so safe to natural enemies. They target juvenile harmful insect populations while causing less harm to beneficial insects. IGRs do not 311. Affect an insect's nervous system, making them more friendly to worker insects in closed environments. Biodegradable, non-persistent and non-polluting. Non-toxic to humans, animals and plants. IGRs are also more compatible with biological pest management systems. Insects can develop resistance to insecticides. They are less likely to develop resistance to IGRs. Disadvantages. Kills only certain stages of pest. Slow mode of action. Because they are chemicals, resistance may develop uncertainty in the environment. Mechanism of action. In order to produce a new exoskeleton underneath its existing one and then shed the old one so that the new one can solidify and swell, an insect molts as it grows. IGRs stop insects from maturing by interfering with the molting process. As a result, infestations are reduced since young insects cannot reproduce. These IGRs kill insects more slowly than conventional pesticides because they prevent an insect from molting properly. Depending on the IGR product, the insect's life stage at the time the product is administered, and how quickly the insect develops, death normally happens within 3 to 10 days. Before some IGRs kill insects, they stop feeding. IGRs stop insects from maturing by interfering with the molting process. Immature insects are unable to reproduce, which lessens infestations. Because they impede the process of molting, these IGRs kill insects more gradually than conventional insecticides. Depending on the IGR product, the insect's life stage at the time the product is administered, and the rate at which the insect develops, death occurs in 3 to 10 days. Some IGRs make insects stop feeding for a long time before they eventually perish. 13.6.1 Juvenoids. 312. Juvenile hormone is also called as neotenine. It is secreted by paired glands present behind insect brain corpora allata. They keep the larva in juvenile condition. JHI, JH2, JH3 and JHIV have been identified in different groups of insects. Juvenile hormone concentration decreases larva reaches the pupal stage. Juvenile hormone I2 and IV are found in larva and juvenile hormone 3 is found in adult of insects. This hormone also helps in development of ovary in adults. Synthetic analogs of juvenile hormone are called juvenoids. They are hormonal insecticides. They sterilization, change the behavior of insect and disrupt diapauses and trigger dormancy. Mode of action. On insect larvae, juvenoids have an anti-metamorphic impact. Insects maintain their current state, the larva remains the larva, and additional supernumerary moltings occur, resulting in superlarva, larval pupal, and pupal adult intermediates, all of which result in the death of insects. Juvenoids interrupt diapause, impair insect development, and have larvicidal and ovicidal effects. As a JH mimic, methoprene is effective against hornfly larvae, pests from stored tobacco, homopterans in greenhouses, red ants, and leaf-mining flies of vegetables and flowers. A high concentration of juvenilizing chemical results in the production of an intermediate larval stage or pupa. 313. Some of the juvenile hormone analogs. Phenoxycarb, Varicil, Insigar, Logic. Hydroprene, Gentrol. Methoprene, Precor, Pyroproxafen, Nigard, Nylar, Sumalarv. 13.6.2 Ecdysoids. Ecdysoids are also called as molting hormone. Its synthetic analogs are called ecdysoids. Ecdysone is secreted by prothoracic glands. They are steroids. Molting in insects possible because of the secretion of ecdysone. In adults its level decreased and it's almost absent. When ecdysoids, the synthetic analog, is applied in insects, is forms defective cuticle, ultimately insect is killed. 314. 13.6.3 Anti-hormones. Anti-JH or precasines obstruct JH synthesis and damage corpora allata. By preventing the generation of juvenile hormone, they counteract its effects. When applied to an insect in its juvenile stage, it skips 1 or 2 larval instars and develops into a tiny, immature adult. Early instars given an anti-juvenile hormone treatment molt into non-functional adults before they should. They are unable to reproduce and will soon pass away. Example, EMD, FMEV, and PB piperenal butoxide. The fact that these chemicals are selective may make it difficult for manufacturers to create them economically. 13.6.4 Chitin inhibitors. Chitin synthesis inhibitors prevent the production of chitin, a carbohydrate necessary for the formation exoskeleton of the insect. With these inhibitors, an insect grows normally until it molts. The inhibitors prevent the new exoskeleton from properly developing, which kills the insect. Depending on the insect, death may occur quickly, or take up to several days. Inhibitors of chitin production can potentially destroy eggs by interfering with proper embryonic development. Chitin synthesis inhibitors affect insects for longer periods of time than hormonal IGRs. Benzoylurea insecticides are among the substances. It has been discovered that benzoyl phenylureas can prevent chitin synthesis from occurring in living organisms by suppressing the action of the chitin synthetase enzyme. Diflubenzurin, dimlin, and penflurin are 2 significant substances in this group. Among the effects they have on insects are molting disruption, labrum and mandible displacement. Inability to break free of the pupal skin results in death for the adult. Chitin synthesis inhibitors have been successfully used against pests of soybean, cotton, apple, fruits, vegetables, forest trees, mosquitoes, and stored grain in numerous countries where they have been approved for use. Chitin synthesis inhibitors. Diflubenzurin, vigilante, a benzoylurea. Triflumarin, stericide, another benzoylurea. 315. 13.7 Summary. Pest management is an important component in management of the pest. Conventional chemical pesticides are harmful as they cause various environmental issues like change in soil fertility, change in aquatic food chain, health issues such as severe cancer, neurological disorders, hormonal disturbances, and reproductive issues, etc. Furthermore, most synthetic pesticides are ineffective as insects develop resistance against them. Botanical pesticides are alternative of conventional pesticides. Example includes pyrethrins, rhodonone, sabadilla, nicotine and neem. Out of this, neem-based pesticides are highly used in India of pest management, followed by pyrethrins.